Right now, some of the most lucrative fields in science, from programming targeted cancer treatments to building quantum computers, face the exact same physical bottleneck. We cannot reliably manufacture at the atomic scale. This graphic illustrates traditional stochastic chemistry. Mixing organic compounds and catalysts in a solution is like shaking a box of Legos, hoping they connect. This high-entropy environment leads to uncontrolled side reactions, giving us a very low yield of usable material. To scale the next generation of technology, we have to stop relying on chance collisions. We must move away from random mixing and build a highly controlled physical assembly line for molecules. The engineering alternative is called positional chemistry. Instead of free-floating chaos, we use a physical molecular scaffolding, a jig, to hold the reactants in place. This gives us a defined reaction path and sub-nanometer positional accuracy. To build these jigs, scientists use a technique called DNA origami. They take long strands of DNA and fold them into custom, rigid 3D shapes, all accomplished at room temperature using a microfluidic chip. This diagram outlines the proof of concept. The folded DNA acts as a highly precise pegboard. By engineering specific sticky ends onto the DNA lattice, it can capture larger molecular components, like these vertically aligned carbon nanotubes, with atomic accuracy. This technology transforms DNA into a physical structural material, a microscopic framework engineered to hold reactants in a precise, fixed position. The immediate goal of building these microscopic factories is to assemble precision carbon nanotube junctions. These tiny, perfectly aligned structures are the critical hardware needed to build functional quantum electronics. This illustration shows the long-term vision of how we get there, scaling from wet biological folding to dry mechanosynthesis. In this hybrid system, the DNA lattice holds the workpiece completely still. Above it, an STM-style tungsten tip provides the activation energy, transferring single atoms directly onto the target. On a computer screen, this hybrid assembly solves the atomic manufacturing bottleneck perfectly. But executing atomic manipulation in the physical world is a completely different story. This comparison shows what happens when perfect theory meets actual physics. On the left is the pristine DNA lattice designed in software. On the right is what environmental entropy, Brownian motion, and surface tension actually do. The structure collapses into an unusable hairball. The environment fights back in other ways. This diagram highlights the threat of nucleases, highly destructive enzymes in ordinary dust and skin that instantly chop expensive DNA scaffolds into useless soup. Even if perfectly sterile, you face microfluidic hell. Look at the right. A single microscopic air bubble trapped inside the channel creates constant friction, permanently blocking flow and ruining the entire chip. Building these microfactories is a brutal fight against thermodynamics. You are trying to assemble the world's smallest puzzle, essentially building a ship in a bottle while wearing oven mitts. This chart highlights the yield crisis blocking commercialization. Academic labs publish papers when only 15% of nanotubes are positioned correctly, but a viable industrial product requires over 80%. To prove you've hit that threshold, scientists use gel electrophoresis. Looking at the bottom right, a broad, noisy smear means the DNA turned into misfolded garbage. A sharp, distinct line, a tight band, is the definitive proof of a high yield of correctly folded structures. Achieving that tight band is the non-negotiable prerequisite. Until you have that visual proof, any money spent trying to scale a full microfactory is entirely wasted. This table breaks down the reality of building an internal lab. What starts as a cheap DIY project cascades into months of delays. As you move down the milestones, costs increase and the probability of success drops to the floor. This image presents the strategic choice facing Synestic systems. Instead of struggling through the messy, high-risk DIY path on the left, the company is opting to outsource the initial physics validation to a specialized contract research organization, shown on the right. This execution plan illustrates why. By outsourcing, Synestics reduces 18 months of operational risk into a highly predictable 36-week schedule. Each phase has guaranteed deliverables, clear costs, and strict kill clauses if the yields aren't met. To solve the obvious risk of handing proprietary molecular designs to outside labs, the company uses strict anti-sneak contracts. As shown in the diagram, all of the foreground IP, The custom sequences, the data, and the protocols remain exclusively the property of Synestex. When the physics are this hostile, attempting to build out your own infrastructure first is a mistake. You have to buy technical certainty from the experts before you scale. Proving the physics of these scaffolds provides a strategic entry point into programmable drug delivery, precision cages that encapsulate chemotherapy and only open in the presence of specific cancer markers. By combining the physical precision of DNA origami with a rigid, milestone-based execution plan, positional chemistry scales out of the laboratory and into the commercial supply chain. If you enjoyed this video, please give a like and subscribe. And what would be really helpful would be some suggestions for future videos. Today's video was brought to you by Synestex Systems LLC,